On Earth, there are two major types of cells, prokaryotic and eukaryotic. Prokaryotic cells are the simplest and most ancient types of cells. They were the only form of life on Earth for billions of years before eventually giving rise to eukaryotic cells. If you take a look at prokaryotic and eukaryotic cells side by side, you can see a number of differences. To begin with, prokaryotic cells are a lot smaller than eukaryotic cells and have a simpler structure. But bear in mind that this simple structure is actually a good thing for prokaryotes because it allows them to reproduce very quickly and very effectively. If you were to look inside a prokaryotic cell, you would probably be surprised at how simple it is. For example, prokaryotic cells lack a nucleus. In fact, the name prokaryote actually gives you a hint about its structure. Kary means kernel or nucleus, and pro means before. So basically the word prokaryote means before nucleus. The inside of the prokaryotic cell is basically an open unit with no compartments. There are no membrane-bound organelles. However, small structures called ribosomes are scattered throughout its cytoplasm. The cell's DNA is located in a region of the cytoplasm called the nucleoid region. The nucleoid region is not the same thing as a nucleus because it's not enclosed by a membrane. Prokaryotes also have a cell wall that surrounds the plasma membrane. The composition of the cell wall depends on whether the cell is an archaean or a bacteria, but basically it consists of complex polysaccharides. Prokaryotes also often possess one or more flagella, which are used for movement. In contrast, eukaryotic cells contain a membrane-bound nucleus and other membrane-bound organelles. The name eukaryote also gives a hint about its structure. Eu means true, and remember that kary means nucleus, so in essence the word eukaryote means true nucleus. Eukaryotic cells are larger and much more complicated than prokaryotic cells. However, they do share a few things in common with prokaryotes. Let's take a look at a Venn diagram to compare prokaryotes to eukaryotes and to also see what areas they have in common. Let's start with their commonalities. Both prokaryotes and eukaryotes have DNA as their genetic material, have ribosomes, have cytoplasm, and have a plasma membrane. Now let's look at some of the ways they differ. Prokaryotic cells are the oldest type of cell. They're small and relatively simple. Eukaryotic cells evolved from prokaryotic cells later. They're larger and much more complex internally. Prokaryotes lack both a nucleus and membrane-bound organelles. Eukaryotic cells have a nucleus and also contain organelles. Prokaryotes are single-celled organisms. Eukaryotes can be either single-celled or multicellular. And finally, the DNA of a prokaryote is usually organized as a single circular chromosome, while the DNA of a eukaryote is organized as linear chromosomes.
So here's our cell ready to start mitosis, right? So it wants to copy its DNA to pass a full set on to each of its daughter cells. So here's our DNA. Remember, in eukaryotes like us humans, we have nice linear strands of DNA. And we have lots of them, as opposed to our prokaryotic bacteria friends who have circular DNA. So here's our linear DNA, and actually at the ends of our DNA strands, right, on both sides, we have these little caps, sort of like the little plastic tips on the ends of your shoelaces. And just like how the little plastic tips protect your shoelaces from fraying or getting worn out, our little telomeres, that's what these tips are called, our telomeres prevent damage from happening to our DNA during the copying process. So to clarify, these telomeres at the ends here don't actually code for any proteins. They don't have any genes on them. All the important DNA is in this section here, this middle section. So I said that there's potential DNA damage during the copying process. What do I mean by that? Well, the machinery that copies our DNA polymerase system, it kind of works in a funny way. It doesn't actually copy our DNA all the way to the end. So what we see is that each time DNA is copied, you lose a little bit of telomere here. So every time the DNA gets copied, you lose a little bit more and a little bit more and a little bit more of the end. So lucky for us, instead of having really, really important coding DNA at the end of our chromosomes, we've got our telomeres. So again, it's the telomeres that end up becoming slightly shorter at the end of each DNA replication cycle. And it turns out that a cell can undergo about 60 or 70 or so cell divisions before the telomeres get too short, and then all this important coding DNA here starts to become at risk of damage from further replications. But not to worry, we have a contingency plan built in. So once the telomeres get too short, the cell initiates a DNA damage response. So it sort of rings these internal alarm bells. And it sort of gives up its ability to divide, because it really wants to prevent any more shortening of this chromosome that would happen in further rounds of replication, right? So it loses its ability to divide, and by doing that, it has become what's called a senescent cell. And before I explain what exactly that is, there's just two things I want to point out about this. So the first thing is that if a cell reaches senescence because its telomeres have become too short, we say it's reached replicative senescence. And the second thing is that the number of times a cell can divide before reaching senescence, replicative senescence, is called its Hayflick limit. And that's named after the scientist who figured that out. And again, it's generally around about 60 or so divisions. So now that we've gotten all that out of the way, what exactly is senescence? Well, basically it's a change in a cell's state from a happy, active, mitotic cell, right, i.e. one that divides on a regular basis, to one that's non-dividing and possibly less happy. I'll have to ask a senescent cell how it's feeling one of these days. But it undergoes all these other changes as well, like it starts expressing all sorts of genes that it didn't before, and it starts to look a little bit different, and it starts to respond a bit differently to the cells around it. But the important thing is that while it's still contributing to the structure of your body, it's still hanging around in your tissues, it's not allowed to replicate anymore. And kind of the key with all roads to cellular senescence, be it through reaching your Hayflick limit, as we just saw, or through other ways, which I'll mention in a second, the key is that cells transform into a senescent state to prevent impending DNA damage from happening and causing maybe cancer or a tumor, or possibly being passed on to daughter cells. So the cell's ability to divide gets turned off. And other causes appear. Well, essentially anything that threatens the integrity of your DNA. So maybe if you had telomeres that just don't work properly, or maybe you sustained some DNA damage through a mutation of some type, or maybe some toxin damaged your DNA, any of these could really just as easily result in your cell becoming senescent, non-dividing. So is there actually a problem with senescent cells? Are we happy about them? Are we upset with them? What's the deal here? Well, we're not really sure yet, but conceptually an easy way I like to think about them is that, well, on one hand, they help prevent tumors or cancers from happening because they're not allowed to divide anymore. But on the other hand, as we get older and our tissues have more and more senescent cells in them, the tissue can't really repair itself as well as it used to because there's fewer actively mitosing cells around, right? And there's actually also some evidence linking senescent cells to age-related diseases like cataracts. But this is a pretty active area of research, so for now we'll just have to keep an eye on these cells to see what new research comes up with for us.